Hello, everyone, and welcome to today's broadcast, Exploiting Cis-Regulatory Enhancers as Therapeutic Targets in Cancer. It is presented by Martin Hirst, PhD, an Associate Professor and Associate Director at the University of British Columbia. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I'd like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. Our speaker will respond to your questions via email. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Dr. Hirst. I will now turn the presentation over to him.
Well, thank you. So let's get started. So as I hope everyone knows, although your genome is encoded in a string that's about 2 meters in length, it's exquisitely packaged into your nucleus, which is a mere few microns, and subjected to both heritable and transient regulatory control. And we understand a number of mechanisms by which this regulatory control is maintained through direct modifications to chromatin. Chromatin being the combination of the DNA, the nucleic acid in your genome, and the proteins that package it. And the proteins that package it are called histones, and these work together in a unit, a complex called the nucleosome. The nucleosome itself, and those are the little balls that you'll see in the slide that you're looking at now. These histones packaged within the nucleosomes have N-terminal tails that are disordered, and these are decorated with chemical modifications that can act to reinforce active and repressed states. There are other mechanisms of regulatory control, including regions of the genome that are open, and these are marked in the presentation by these so-called DNase 1 hypersensitive sites. And there are modifications directly to the DNA itself. This is primarily at least in mammalian somatic cells in the context of CPGs. This is DNA methylation, so a methyl group added to the CPG, but it can also be a hydroxymethyl and other oxidative forms. We know that RNA, which is also indicated in the slide, plays a role in regulating the epigenetic code through interactions with the enzymes which act to lay down these modifications. Now, just to get a little bit more detail in terms of what we understand about these epigenetic modifications, of course, DNA methylation has been known, was actually initially discovered only 5 years after after the famous experiments of Avery, McLeod, and McCartney determined the DNA to be the hereditary agent, but it took decades of work actually to understand the significant role that DNA methylation plays in differentiation of cells. So what we understand about DNA methylation today is that CPGs, these are, as I mentioned, the area within, or the sequence within the mammalian genome that is methylated. CPGs are not randomly distributed in the genome, rather they're collected in areas that we call CPG islands or CPG-rich regions. But actually, these CPG-rich regions represent the ancestral CPG content of the genome. And over the eons, through spontaneous deamination, other CGs have been lost. But these CPG islands have been maintained, presumably under selective pressure, because they act in a regulatory way to control the expression of genes. So CPGs are unevenly distributed in mammalian genomes. And actually, if you look at a normal somatic cell, the majority of CPGs in your cell are actually methylated. but we do see a specific decrease correlated with differentiation, with cellular differentiation, and also in specific cellular contexts. We now understand that gene bodies themselves are methylated, and this is something that is relatively recently been determined, or at least the mechanism has relatively recently been determined. And traditionally, or at least dogmatically, we've considered methylation to be associated with gene repression, and this is true, but only in very specific contexts. And if you look at the figure at the bottom of the slide, you'll see some specific patterns of DNA methylation that I just mentioned. So, for example, within an inactive gene body, we see DNA methylation, and with enhancers, we see a hypomethylation or a loss of methylation, and then we also see this loss of methylation within transcription factor binding sites, which we'll talk about in a little bit more detail as we go through this presentation today. Now, in addition to DNA methylation, as I mentioned, histones themselves are also modified, and we know of over 100 different modifications that can occur. Advances in technology over the last decade have enabled us to actually measure these epigenetic features genome-wide using technologies such as ChIP sequencing, so chromatin immunoprecipitation sequencing. And a number of consortia have used these technologies to map these marks genome-wide. One of the first of which was the NIH Roadmap Consortia, who defined a so-called reference core set of histone modifications, which I'm showing on the right-hand side of the slide. And these include modifications that reinforce both active as well as repressive states, and other modifications that are found associated with enhancer genes, as is indicated on the right-hand side of the slide. And just as a reference, the way that the nomenclature that's developed for referring to histone modifications is shown there. So the first two letters indicate the histone, in this case, histone H3. And then we have the specific amino acid that's modified, so lysine 4, so K, lysine 4 is the fourth amino acid from the N-terminal tail, remembering that the N-terminal tail is this unstructured tail that sticks out of the nucleosome, as I showed earlier. And then M3 indicates the modification, in this case, 3-methyl groups. And what you can see is that, in fact, different types of modifications occur at the same amino acid. So, for example, H3K27 acetylation, mark of an active enhancer, but it can also be trimethylated, as is shown at the bottom, and this is indicative of a repressive mark. So both active and repressive chromatin states exist, and these can also be, and these are also used to discover and to annotate other regulatory features in the genome. Okay, so unlike your genome, which is largely the same throughout an organism's life, and in fact is also largely the same throughout all the cells in your body, with the exception of some specialized immune cells, the epigenome is distinct, and it's distinct in all of the cell types of your body. So as far as we have determined, each cell type, each unique cell type in your body contains a unique epigenetic state. And so you can imagine that this is much more challenging to determine what these unique epigenetic states are throughout all the cell types in your body. And even more complex is that we know that the epigenome can be modified by environmental stimulus. So different environmental stimulus can change the epigenetic signature, and thus it's a dynamic interface between the environment and your genes, and it is also plays a significant role in specifying cell type. So as I mentioned, there are a number of consortia who are working together using technology, again, that's been developed in the last decade to begin to try and generate a series of what we call reference epigenomes, which are meant as a foundational resource, much like the reference human genome, to allow us to begin to understand what are the patterns associated, or what are the epigenetic patterns associated with a given cell type, and how do these epigenetic patterns change as a consequence of disease. One such consortia is the Canadian Epigenetics Environment and Health Research Consortium, which is a member of the International Human Epigenome Consortium. The types of data we generate are shown in this matrix, data matrix here. This is something those of you who are familiar with, for example, ENCODE or the NIH reference mapping activities would recognize this type of matrix. And here we can see along the top the types of modifications that are, or the type of epigenetic states that are measured. And then along the rows, we can see the particular cell types that have been assayed. And so really the goal here is to fill these matrices for all of the cell types, all of the normal cell types in the human body as a reference, again, to then understand their dysregulation in disease. And I'm making the arrow indicates a particular reference that's been generated, in this case for a well-studied mammary cell line, MCF10A. This is a fibrocytic cell line. And I'm just making the point in the next slide that along with these reference epigenomes, we have generated a collection of standardized both sample, experiment, and run metadata that are available through the public repositories for researchers such as yourself to then parse this information out and download these full reference epigenomes for your own research purposes. And again, in the next slide, just to make the point that as part of this process, we also link all of the samples to ontologies, and these ontologies then allow you to do large-scale analysis across these reference epigenomes. So collectively, groups globally are now working together as part of the International Human Epigenome Consortium to generate these reference maps. As I mentioned, one of the founding members is the NIH Roadmap program. ENCODE is a member, 40N, as well as the CIRC network, and members across Europe and in Asia. And again, the purpose of this consortia is really to generate these reference maps that can then be used for studying diseases. These reference maps are published through public repositories, but are also available or have been published in these collections of manuscripts, first within a Nature compendium, shown on the left, and more recently within a package of cell papers. And currently the next iteration is currently being worked on. So with that sort of primer in terms of what we understand about epigenetics and our activities around generating references, what do we understand about epigenetic dysfunction and disease? And in particular, I'm going to focus in today's talk on cancer. So one of the interesting findings that's emerged after the last decade of sequencing cancer genomes, of course, using the same technology that is allowing us to measure epigenetic features, has also been applied to study cancer genomes. And one of the most surprising findings was that the finding that there are across many types of cancers, there are recurrent gain and loss of function mutations to epigenetic modifiers. And these can be really classified in terms of their frequency. So for example, in some cases, these mutations are pathognomonic. They're actually describe the disease. For example, in a pediatric malignant rhabdoid tumor, almost 100% of these tumors contain biallelic loss or mutations which drive biallelic loss of an epigenetic modifier called SMARCB1. They're recurrent, so we see them at very high frequencies in some tumors. For example, in glioma, we see gain of function IDH mutations in nearly 80% of low-grade glioma. And when we look, and now we can look at very high resolution at pretumorous lesions, we can see that these epigenetic modifying mutations are very early events. And I'll talk a little bit more about that as I go through the presentation. So just to give a few examples of what I'm talking about. So first of all, rhabdoid tumors, so malignant rhabdoid tumors, these are really defined. So it's really a pathognomonic description or a pathognomonic mutation. And these are really defined by the biallelic loss of SMARCB1. And what I'm showing you here is really just a compendium of data that's been generated across a cohort of 47 malignant rhabdoid tumors And what you can see is that on the top row is that the only commonality here, again, is loss of SMARCB1, with the sole exception of one tumor that actually has a loss of SMARCA4, which is another member of the same complex, in this case, the SWI/SNF complex. This is a very aggressive tumor. It happens early in life, usually before 1 year of age, and is almost universally lethal. So here we have an example of a mutation to an epigenetic modifier that drives a very specific disease in a very specific context. Another type of tumor that appears to be driven by epigenetic dysfunction is again a pediatric tumor, in this case ependymoma. And this particular tumor is actually the only tumor that at least I'm aware of that appears to not have any genetic mutations to epigenetic modifiers, but rather appears to just have evidence of an epigenetic alteration. So on the left-hand side, we can see a panel which shows the mutational frequencies across a series of different tumor types, with ependymoma being at the very bottom. And what you can see is there's essentially no recurrent mutations that can be found in this particular tumor type. And then on the right-hand side, what we can see, though, is that there is a clear signature, a so-called CIMP signature. So this is a CpG island methylated phenotype, as it's called, where we see a clear gain of methylation within CpG islands in a subset of ependymoma cases. So again, no evidence of genetic lesions, but evidence of epigenetic alterations associated with disease. And as far as I'm aware, this is the only tumor type of this class. Another class of tumors, and again, we're staying within pediatric malignancies. This is really pediatric CNS tumors, where we see these mutations, not to the epigenetic modifiers, but rather to the N-terminal tails themselves. So these are the tails, the unstructured tails that are decorated with these epigenetic modifications. And in fact, we can see mutations directly to the amino acids that are modified. And these are highly specific. So we see specific mutations to specific amino acids associated with a subtype of cancer, the subtype of a pediatric CNS tumor, which emerges in a particular area of the brain. So very highly specific tumors early in life, and again involve some epigenetic dysfunction. However, in this case, this is through mutations to the N-terminal tails of the histones themselves. So another example is low-grade glioma. And low-grade glioma are associated with neomorphic, so gain-of-function mutations. In this particular case, gain-of-function mutations to IDH1. And in this study, published in 2014, really highlights the feature of these early events that I mentioned earlier. So even when we look in normal tissue that's adjacent to the tumor itself, what we see is mutations, this gain-of-function mutations in IDH1. And I'll be talking a little bit more about this. I'll be talking a lot more about this particular mutation as we go through the slides. But suffice it to say that this mutation leads to epigenetic dysfunction, and it's found both early, and then if we look along these trees, so on the left-hand side and the right-hand side, there are two different patients where we've tracked the mutations that occur in each of these tumors. And what you can see is that these mutations are very early, so they're in the blue bars, you can see they're actually present in the adjacent normal tissue. But then also, as we look through the history or as the tumor has evolved, these IDH mutations are in fact ubiquitous. So they're found in all of the recurrence of the tumor and also very early in the tumor progression. So finally, another type of tumor, in this case, we're talking about a liquid tumor, and in this case, acute myeloid leukemias. So acute myeloid leukemias are associated with a large number of mutations to epigenetic modifiers. And in fact, if we just look at the class of mutations that affect DNA methylation, and those are the ones that I boxed in red in this figure, we can see that nearly 44% of de novo AMLs contain a mutation to one or more enzyme that controls DNA methylation homeostasis. So this includes in the top row, and again, looking at this figure, the rows in this particular instance indicate genes, and the columns are individual patients, and then the colors indicate where a mutation has occurred. And so what you can see is that if you look in the red boxed area, you can see a series of enzymes. The first three are the DMTs. So these are the enzymes that are responsible for laying down methylation. So these are the methyltransferases. The first two being the de novo, and then we have the maintenance. And then we have the TET enzymes, TET1 and 2, and these are enzymes that are responsible for removing methylation. So they involved in hydroxymethylation, and then either through active or passive mechanisms leads to the DNA demethylation. And finally, again, the IDH enzymes, and again, these are the same gain-of-function mutations that we see associated with glioma. And as you'll see, these IDH mutations actually act upon TET2. One other point I'd like to point out is that when we do see these TET or IDH mutations, these are mutually exclusive. In other words, we don't see them together in the context of acute myeloid leukemias. So just to kind of reiterate that point and give a little bit more detail, in the next slide now I'm showing you the mechanism by which DNA methylation homeostasis is maintained. So you can see on the left-hand side, we have DNA that's methylated. So you can see a cytosine that's methylated, and TET2 through alpha-ketoglutarate then hydroxymethylates the cytosine. And then either through an active mechanism, through basic excision repair, or through a passive mechanism, in other words, through cellular differentiation, the methylation that was initially present and shown on the left-hand side is then lost. So TET2 and the TET family, so there's three, TET1, TET2, and TET3, are responsible for removing methylation in the genome. Now, the gain-of-function mutation to IDH generates, instead of generating alpha-ketoglutarate, generates an oncometabolite called 2-hydroxyglutarate. And 2-hydroxyglutarate acts upon the TET enzymes and actually inhibits them. So the gain-of-function mutation of IDH phenocopies the mutation to TET2 or the loss-of-function mutation to TET2, leading to its inhibition. And as I just mentioned, in the context of acute myeloid leukemias, we see either TET2 mutation or IDH. We don't see them together. And curiously, for reasons that we don't fully understand, in the context of gliomas, we only see the IDH mutation. And just to finally mention that the 2-hydroxyglutarate is also capable, is also capable of inhibiting lysine demethylases. So the way that we remove methyl groups from histones, as is indicated at the top of this figure, also goes through a hydroxymethylation step via lysine demethylases, and 2-hydroxyglutarate has been shown to inhibit these as well. Okay, so the TET enzymes, as I just introduced, are actually part of a family of 2 oxoglutarate-dependent deoxygenases. And I'm showing in this slide, I'm showing you a phylogen or a dendrogram of the 2 oxoglutarate-dependent deoxygenases. And you can see in the blue color here, there's the TET, so these are the nucleotide hydroxylases. And then you can see in the yellow and the green and the purple, these are all the histone demethylases. So these are the KDMs, the Jumanjis, for example. And then in the gray, we can see some proline hydroxylases. So these are another class of hydroxylases encoded in the genome. Now, what we know is, or one hydroxylase that is perhaps well known is actually the causative agent of scurvy, so-called proline hydroxylase. And proline hydroxylase, which is shown on the right of this figure, is responsible for hydroxylating collagen, and this hydroxyl form is required to form collagen fibers. And of course, in the absence of vitamin C being an essential nutrient in humans, or in humans and primates, in the absence of vitamin C, this hydroxymethylation does not occur, and you get collagen breakdown. So what vitamin C is required for this reaction and it acts to reduce iron, and this reduction is required for active proline hydroxylation. So given this, and given that TET is a member of the same family, we asked some years ago whether it would be possible for vitamin C to also act upon other deoxygenases, for example, the TET family. And so what's being shown in this figure in panel A is immunohistochemistry of, in this case, murine embryonic stem cells following vitamin C treatment in culture, and then doing immunohistochemistry against hydroxymethylcytosine. And what you can see is that after 12 hours of treatment, you can see a large gain, or a gain in hydroxymethylation is indicated in the signal. If you look on the lower panel, what is shown here is actually a quantification of this change genome-wide, and now we're looking at a particular gene here. And what we can see is that there's actually a dynamic response to the addition of vitamin C. So starting in the untreated, at the top, you can see hydroxymethylation at 12 hours with the blue density of the blue track. And what you can see is that after 12 hours, you get a gain in hydroxymethylation. And then in the maroon tracks underneath, you can see that in the untreated state, in fact, this gene is heavily methylated. The CGs are shown at the very bottom. Those are the slashes, the black slashes. But you can see that this gene is heavily methylated when it's untreated. And then after 72 hours becomes unmethylated. So putting this together, what we observed is that following vitamin C treatment, we see a gain of hydroxymethylation at the gene, and this leads to a subsequent DNA demethylation event that occurs at 72 hours. So consistent with a model where vitamin C might be activating the TET enzyme, leading to this hydroxymethylation event. And then the dot plot on the right-hand side just shows this again in a quantitative measure, showing the gain of hydroxymethylation following vitamin C treatment. So I've already told you that vitamin C is required for reduction of iron, which is required for the 2-hydroxyglutarate-dependent deoxygenases. However, there's also some evidence in vitro for direct interaction between vitamin C and the TET enzymes. So again, looking at the top panel, you can see that when we add vitamin C to media, or when we add vitamin C in vitro, we can see activation, in this case of a proline hydroxylase. This was data generated many years ago, and this was then replicated below, in this case looking at TET2, and we can see that by adding, so this is the lower panel on the left side, by adding vitamin C, we can see induction in TET2 activity. Whereas if we add other reducing agents such as DTT or glutathione, we don't see that activity. Again, suggesting that perhaps there's something distinct about vitamin C's ability to activate TET2. And indeed, around the same time that our paper was published, another group showed that using an intrinsic fluorescence measurement, that there appears or suggests that there might be a direct interaction between vitamin C and TET2. So you can see on the right panel that vitamin C addition induces a change in this intrinsic fluorescence, which is suggestive of a direct interaction perhaps between vitamin C and TET2. So what do we know about vitamin C in terms of its activity or its distribution in the body? Well, of course, it's been studied for many years and has been measured in blood serum. And looking at the top left-hand side, this is a panel looking at, or this is a panel showing ascorbic acid in the blood. And what we know is that ascorbic acid or vitamin C's levels in the plasma is tightly regulated. So on the x-axis, we can see a dose per day, so the or dose milligrams per day. And then on the y-axis, we can see the plasma concentration in micromoles. What you can see is that you quickly plateau, and then just increasing the amounts of more vitamin C, at least orally, doesn't lead to any increase in the plasma concentrations. In fact, the additional vitamin C is simply excreted. Now, what's curious, and on the right-hand panel at the top, what I'm showing you is a publication from 2011, which showed that actually when we look within monocytes and other blood cell types, we actually see an increased concentration of vitamin C. So now going from through low nanomole or high nanomole to micromole, sorry, from high micromole to millimolar concentrations. So we can actually see that within monocytes, we're actually reaching between 3 and 4 millimole concentrations of vitamin C. Again, suggesting that vitamin C is being actively pumped into these cell types and actually being concentrated. This was recently confirmed and then extended by Sean Morrison's group, publishing last fall, showing that if you actually break down the blood cell types into hemopoietic progenitor cell types, such as HSCs and multi-progenitor cell types, you actually see that the increase in vitamin C concentration is associated with these progenitor stem cell like cells. So this is shown on the bottom left panel. And on the right panel, what you can see is the concentration of ascorbate within these cells is actually directly correlated, not surprisingly, to the expression of a transporter that transport vitamin C. And so you can see again in the top right-hand quadrant of the bottom right figure, you can see this gain of vitamin C in the hemopoietic progenitor population. So taking this all together, we asked whether, or we conjectured, well, maybe, given what I told you about TET inactivation in the context of acute myeloid leukemia, either through gain of function mutation to IDH, which directly inhibits TET2, or through a direct genetic mutation to TET2, we asked, or we conjectured maybe, or could vitamin C act to overcome this inhibition and directly reverse epigenetic alterations that are associated with these, either the gain of function or loss of function mutations, gain of function IDH or loss of function mutations to TET2. Looking back in the literature, we can see that in the late 80s or early 90s, a study actually looked at the consequence of adding ascorbic acid to cultures. So either looking at normal hemopoietic cell types or myelodysplastic cells. These are a sort of a pre-leukemic state or can lead to frank transformation. And then looking at at leukemic blasts and asking what is the consequence of adding ascorbic acid to the media. And what you can see is that in many cases, and that's along the dotted line here, there was really no change in the cell, but there was a set of leukemic cells that responded to the ascorbic acid. And one might conjecture that the ones that responded, it may in fact be that subset of leukemic cells that contain inactivating mutations to TET or gain of function mutations to IDH. So we set out to directly test this, in this case using a leukemic model. In this model, we take bone marrow cells from a mouse and we immortalize them using a well-known oncogene, HOXA9, and that's shown. So in the left-hand panel, we immortalize them using HOXA9, and then we add to these immortalized cells either wild-type IDH, so this is just the IDH wild-type form, or a gain-of-function mutation. This is indicated here as IDH mut. And on the left-hand panel, so panel A, we can show that when we add the HOXA9 IDH mutation, we indeed can see production of 2-hydroxyglutarate. Again, this is the oncometabolite that inhibits TET2. And then on the right-hand panel, on the far right panel, panel C, we can see that indeed by introducing IDH mutants into a HOXA9 background or HOXA9 transformed bone marrow cells, we can see that it leads to a much reduced latency into leukemic transformations in the mouse model. So we asked whether we could, by simply adding vitamin C in the context of this model, could we indeed induce hydroxymethylation? And indeed we can. In the top panel, which is probably hard to see, but you can see an immunohistochemistry either using DAPI or using an antibody against 5-hydroxymethylcytosine. You can see an increase after 12 hours of addition of vitamin C. And in the lower panel, this is quantified as a percent of HMC positive cells. And you can see that indeed we see a significant gain in hydroxymethylation in the context of this model through the addition of vitamin C. We then went on to show whether this gain of hydroxymethylation was in fact dependent upon TET, and we did that using a TET2 siRNA. And this is work that was published last summer in Leukemia. And on the lower right-hand panel, this is a student in my lab, Matthew Minge, who did the majority of this work. And what we can see is that by adding this CIHRNA, we in fact see a statistical reduction in the amount of gain as measured by mean fluorescence by immunohistochemistry of hydroxymethylation. So we show that at least this gain of hydroxymethylation is at least in part dependent upon the presence of TET2. So we then went on to confirm this result further by generating a CRISPR TET2 knockout, so knocking out exon 1 of TET2. And again, what we can see, so on this panel, you can see the light gray bar is the control. So this is the HOXA9 gain of function R132H IDH mutation. And then we can see by addition of vitamin C, we see a gain of intensity, that's the dark gray bar. And then by knocking out the exon 1, we can see that that gain is largely lost. Again, suggesting that at least in part, this gain of hydroxymethylation is being mediated directly through TET2. And then the further bars along in this figure show the consequence of adding other DNA demethylation agents in combination with vitamin C. So this includes 5-azacitidine, currently a current treatment of late-stage AML, as well as DAC, a deoxy form of 5-azacitidine. So what happens to the cells themselves in the presence of vitamin C? So on the left-hand panel here, we can see on the top, this is HOXA9 IDH wild type, grown in either the presence of control or in the presence of the carrier or in the presence of vitamin C. And what we can see is that there's very little change. However, when we look at the HOXA9 R132H, we can see that we see cytostatic behavior. And in fact, when we look, as shown on the right-hand panel, what we can see is evidence of differentiation. So looking at the right-hand panel, the lower right quadrant, we can see what appears to be granular cells or granulocytes forming when we add vitamin C in the context of the IDH R132H HOXA9 model. So we next looked at the molecular signature associated with addition of vitamin C. And indeed, as we had seen in the, as was evident in the previous slide, we can see when we try to quantitate the degree of differentiation, as is shown in panel C here, we can see that we see a loss of progenitor markers such as SCA1, and then a gain of differentiation markers MAC1 and GR1, suggesting that addition of vitamin C is indeed providing a push towards differentiation. In panel D, we can show the differential gene expression following vitamin C treatment, and we can see that we see a gain of expression, so we see more genes being expressed as a consequence of vitamin C addition. And when we look at what those cells, what pathway those genes that are being upregulated fall into, you can see that there are pathways that are highly relevant to differentiation, leukocyte regulation, and leukocyte differentiation. Again, suggesting that the addition of vitamin C is specifically driving a differentiation of this particular model. So we next then looked in the genome to see what epigenetic state changes were occurring. And what we could see is that indeed we could see gains of hydroxymethylation as a consequence of vitamin C, as we had shown with our immunohistochemistry. And we could see that these gains of hydroxymethylation occurred specifically at regions that had gained methylation as a consequence of transformation of IDH R132H. So on the left-hand panel here, what we're showing is regions that are, these are the regions that gained methylation so we have our control with a wild type IDH, and then we have our IDH R132H, which results in a gain of methylation. And when we look at those particular regions, we can see by adding vitamin C, we see this gain of hydroxymethylation, specifically at those regions that gained methylation as a consequence of transformation with IDH R132H. When we look at the genes that are associated with the regions in the genome, that are associated with the regions in the genome that show this gain of hydroxymethylation, these all, again, are associated with genes that are involved in hemostasis, involved in myeloid cell differentiation, again suggesting that this change in methylation is leading to a drive towards differentiation. So we next looked at the genetic signature, or the motif, that was associated with these regions that gained hydroxymethylation and then lost methylation. And what we saw is that these were enriched, or highly enriched, with motifs that bound transcription factors that are known to be involved in mediating or involved in drive or key transcriptional regulators of hematopoietic differentiation, including PU1, which is shown at the top on the left panel, and RUNX1. And it should be noted that PU1 has previously been reported to actually interact directly with TET, and in particular with TET2. As I mentioned, there are 3 TET family members, TET1, TET2, and TET3, within our model and also within primary AML. Only TET2 and TET3 are highly expressed. And TET2 itself is the only one of the 3 TET family members that doesn't have its own DNA binding domain. So it's lacking the CXXC domain, which is used by TET1 and TET3 to be recruited to DNA. And so, but what has been shown is that TET2 actually physically interacts and is recruited to DNA via interactions with transcription factors, as I'm showing in this cartoon on the right-hand side. So PU1 is recruited via this motif, and then TET2, again, interacts directly, leading to the hydroxymethylation of that particular element. When we look genome-wide, so now knowing that we have this relationship between PU1, transcription factor, and the regions that gain hydroxymethylation following vitamin C, we repeated that experiment, but this time we did ChIP-seq against the PU1 itself. And what we can see is that when we do ChIP-seq, we can see that we see a decreased binding of PU1 following vitamin C treatment. And this is enriched in regions that were induced or that lost DNA methylation following vitamin C treatment. And so what we're showing here is that in the untreated state, you can see that there's significantly more PU1 binding sites following vitamin C treatment. There are some that are maintained, and then there's only a very small fraction, only 1.8% shown here, that are unique to the vitamin C treatment, suggesting that perhaps maybe PU1 is being recruited to its methylated element with more affinity than it is to the hydroxymethylated element. We see the exact opposite when we look at RUNX1, and RUNX1 is a transcription factor whose binding is inhibited by methylation. And indeed, what we see is that we see an increase in RUNX binding following vitamin C treatment. So again, looking at the left-hand panel here, these are regions in the genome that are bound by PU1, bound by RUNX1 by ChIP-seq. And what you can see is that we see a large gain of RUNX1 binding following vitamin C treatment, suggesting perhaps that the vitamin C leading to hydroxymethylation, or the binding of PU1, recruiting TET2, leading to hydroxymethylation, is then allowing for RUNX1 recruitment. And so this is the model that describes that. So again, at the top here, we see PU1, which is bound to methylated DNA and recruits TET2. And in the presence of this gain of function IDH R132H mutation, shown on the left, on the top left-hand side of the slide, leads to the inhibition of TET2, and then the pro-differentiation genes are then not transcribed. By adding vitamin C, we're overcoming this block, leading to the hydroxymethylation of these methylated regions. And then this is leading to the DNA demethylation, which is allowing RUNX1 to be recruited to these elements, which is then driving transcription of the pro-differentiation genes that I showed you in the gene expression analysis. And so this is the model which we're currently trying to validate in the context of human PDX models. Now, after we published our paper, two papers were published which showed similar features, but in this case, looking at TET2. So instead of looking in the context of a gain-of-function IDH mutation, looking in the context of direct TET2 inactivation, and showing that TET2 inactivation blocks self-renewal and leukemic progression, and that ascorbic acid is actually involved in regulating hematopoietic stem cell function and leukemia genesis. So in this particular figure taken from the second paper, what is being shown here is another mouse model, but in this case, instead of using the HOXA9 model or HOXA9 oncogene, using the FLT3 IDT. So this is another known recurrent mutation that occurs in the context of de novo AML and is associated with transformation in the context of either a TET2 inactivating mutation or IDH. But in this case, when we have a TET2 inactivation with IDH, and we introduce that into a mouse that has lost the ability to generate vitamin C. So most rodents can generate vitamin C de novo, but we can generate a rodent line, a gluonegative rodent, a gluonegative mouse that is incapable of generating vitamin C. And when we introduce this leukemic model or these leukemic cells into the gluonegative mouse, as is shown in panel B here, you can see that you get this dramatic reduction in latency in the context of the loss of vitamin C. So this is consistent with what we had showed in the context of the HOXA9 IDH R132 model. Around the same time, another paper was published by Peter Jones's group, which actually looked at vitamin C levels in patients who were suffering from a hematopoietic malignancy, and was surprised to find actually that a majority of patients who were undergoing treatment were actually shown to be vitamin C deficient. So that's shown in the top panel here, shown in panel A and panel B, basically showing that looking in patients with these hematologic malignancies, actually that there was this reduction in vitamin C in the plasma. And this finding in its own right has led to a clinical trial, which is actually looking at just oral supplementation in the context of 5-azacitidine treatment in acute myeloid leukemic patients. In the lower panel here, I'm reporting the results of another study, which has looked in elderly or patient subjects who are asymptomatic, but who contain or who looking at genetic mutations in their blood, so essentially deeply sequencing genomic DNA extracted from their blood, actually show these mutations again to the IDH or TET enzymes at some frequency. So these are elderly patients who have essentially asymptomatic, but have evidence of low frequency mutations to either IDH or TET2. And in fact, these low frequency mutations in the context of IDH and TET2 have been shown to be associated with clonal hematopoiesis which is it's also associated with aging, suggesting, like in the glioma model, that these mutations that are occurring to these epigenetic modifiers are actually occurring very early. They occur as very early events, perhaps setting the stage for further transformation. So some questions that are arising from this work is, can sufficient vitamin C in its own right delay clonal hematopoiesis progression? So in the context of those elderly patients that have these mutations, perhaps just sufficient vitamin C would delay the progression from clonal hematopoiesis to either MDS or AML. And finally, or can vitamin C in the context of 5-azacitidine, again, this is a standard of care for MDS and refractory AML, can vitamin C act synergistically with 5-azacitidine to lead to increased DNA demethylation and therapeutic opportunities? And so, as I mentioned earlier, of course, 5-azacitidine acts as a non-specific DNA inhibitor of DNA methyltransferases, so maybe the additional vitamin C can act synergistically. And in fact, in the context of our own work, we're now starting to actually monitor vitamin C levels in patients who are undergoing 5-azacitidine treatment locally to see whether we can replicate this loss of, or this reduction in vitamin C in the context of hematopoietic malignancies. So finally, just to summarize what I have said, so vitamin C treatment is cytostatic and induces hypomethylation in these regulatory regions. So these are regions within the genome that are bound by transcription factors, and also within CPG island flanks in the context of the IDH1 R132H model. And we see transcriptional and regulatory state changes associated with vitamin C treatment, and these changes suggest a reactivation of a myeloid differentiation program in the context of acute myeloid leukemia, and that vitamin C appears to activate specific 2-hydroxyglutarate-dependent deoxygenases, including TET, as I showed. And then finally, that vitamin C may provide a model for a new class of epigenetic therapeutics. So finally, I'd just like to thank the individuals who have done the work, and also the funding that allowed this work to be done. As I mentioned, Matthew Mingay drove the majority of the work that I talked about, as well as many other folks in the lab. And I'd also like to thank Michael Huesler for the mouse model, and Connie Ease and Keith Humphreys, as well as the group at the Genome Sciences Center who have been involved in generating the reference maps, which I introduced in my talk. And with that, I'd be happy to answer any questions.
Thank you, Dr. Hirst, for that informative presentation. We will now start the Q&A portion of the webinar, and we'll address some of the most commonly asked questions by our viewers. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window, type your questions into the box that appears on the screen, and click the Send button. Our speaker will follow up with your questions via email. Let's get started. Our first question is, would vitamin C be effective in other cancers which harbor gain-of-function IDH mutations?
Yeah, so this is an excellent question. And of course, one such cancer would be glioma, which is another cancer subtype where we see highly recurrent gain-of-function IDH mutations. And in fact, we've looked to see in a glioma model whether vitamin C in culture acts to or drives a gain of hydroxymethylation, and indeed we do see that. It's also interesting to note that glioma and AML are both show this the same deregulation of their epigenome, so they both show this gain of methylation at CPG islands, which is associated with the presence of this IDH mutation. So indeed, it suggests that perhaps vitamin C might be might have some therapeutic, action in the context of gliomas.
Thank you. Next one is, why are somatic mutations to epigenetic modifiers so specific in cancer subtypes?
Yeah, so this is an excellent question, and I think is really one that the field is really grappling with. So, I introduced a series of different types of epigenetic modifications, so we can talk about, for example, loss of SMARCB1 in the context of malignant rhabdoid tumors. So this particular mutation gives rise to a tumor type that's very specific to for example, in the context of MRT, to soft tissues like the kidney. And it arises around the same age in all of these patients and is highly specific. We don't see this type of mutation, or at least we don't see this biallelic loss in any other subtype of cancer, suggesting that it's highly specific. And I think that the current sort of thoughts on this is that the epigenetic state of the cell of origin in which this genetic mutation occurs is very specific, and that epigenetic state is what allows for, or when that mutation occurs, then drives the malignancy. And so the specificity is probably, or is likely, a consequence of both the cell of origin when this particular genetic mutation occurs, and the particular genetic mutation itself, leading to the types of specificities that we see. Although really this is an active area of research, and many of us are trying to understand what role the cell of origin plays, or even what the cell of origin is. Just to mention that for many of the tumors that I just introduced, the cell of origin is not known. It's not known for malignant rhabdoid tumors, it's not known for ependymoma, and it's certainly not known for any of the other SWI/SNP mutant tumors.
Oh, thank you for that. We have time for one more question. Are there examples of purely epigenetically driven cancers?
That's an excellent question. So, of course, the reason, I think in the public sort of understanding of epigenetics, I think one of the most exciting things is this concept of interaction between the environment, that somehow your environment can be encoded with your epigenome, and this might then be inherited, even transgenerationally. But it is interesting to note that even though we find lots of evidence, and there are many kinds of cancers that have genetic mutations to epigenetic modifiers, there's only one, and in this case, and it's the one that I introduced, ependymoma, is the only case of a cancer that appears to have no recurrent mutations to genetic modifiers. Now, maybe we just haven't seen it yet, but to date, there really is no evidence that purely epigenetic changes can drive a malignancy. And so, again, it may be that we haven't looked in the right places, or simply that this is a feature of epigenetic deregulation that doesn't in fact lead to transformation.
Thank you. I would like to once again thank Dr. Hirst for his presentation. I'd also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through January 11th, 2019. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.